We will review different types of cell movements during gastrulation, and the purpose of these movements is to establish specific germ layers. In some organisms there are two germ layers, and in humans and in most of the organisms there will be three germ layers, and the ultimate purpose of these gastrulatory movements is to establish these three germ layers. And the type of cell movements during gastrulation have been classified on the basis of three criteria. The first one is direction of movement, whether it is inward movement or it is outward movement of the embryo. And second one is what morphological outcome will it produce, like will there be spreading of sheets, will there be specific type of inward folding, will there be specific type of migration, and what type of layers will it be generating, whether it will be ectoderm and endoderm or it will be ectoderm, mesoderm and endoderm. And the third one is the relative position of moving cells, whether the cells are moving over the surface level only or whether the cells are penetrating deep inside the embryo. So these are the main criteria for classifying these cellular movements during gastrulation. Now, the cell movements can be classified into two main types. If the cell movements are towards the external surface, if the external surface of embryo is involved in these movements and movements are external only, we call it epiboly. And if these cell movements are towards the interior side of embryo, the cells are moving toward the interior of this embryo, we call that emboly. So we have two main types of cell movements. We have epiboly and we have emboly. Now this emboly can be further classified into several types of movements, like it can be infolding or in-punching, which we call invagination. It can be moving of cells, the rolling around of cells with one static layer or static outer layer, but the movement will be toward the internal side, so it will be involution. Or there may be specific cells which will lose their cell adhesion molecular interactions and they start migrating inwards. The individual cells will migrate, we call that ingression. And if a sheet of cells, a epithelial sheet will migrate, we call that delamination. Now we will start looking at these processes one by one. Let's start with epiboly. So epiboly represents that the outer sheet or the outer epithelial sheet will start enclosing the embryo. As you can see, this is the outermost epithelial sheet and the cellular movements are in this direction and it will eventually enclose or uncover this whole embryo. As you can see here in this diagram, this is the outermost epithelial sheet, and after some time this epithelial sheet is covering the whole embryo except this specific region which we call yolk plug. Now there are three main types of movements or three main type of processes which are responsible for this epiboly. The first is cell division. The cells will start dividing mitotically. The second one is change in shell shape. There will be some cytoskeletal changes, there may be some signaling induced changes, and third one is radial intercalations, which means the cells of different adjacent layers will start mixing together to thin the actual layer surface so that the spreading will take place and the whole embryo will be uncovered by this outermost layer, and all these three processes will occur simultaneously. Where does this epiboly process take place? The epiboly process is most commonly seen in case of sea urchins, which are echinoderms. It may be seen in some tunicates, in some amphibians at some stages, and zebrafish. We will take the example of zebrafish when we will study this process in great details. Now we will quickly review the process of epiboly in case of zebrafish. In case of zebrafish, the outermost layer, which is engulfing layer, which is represented by blue color, this layer will uncover the whole embryo externally, as you can see, and the region which is not covered by this layer, we can call it as yolk plug. Now there are two more components which are responsible for this movement of external layer and uncovering of this whole embryo. The one is deep cells, which is represented by DCs, and another one is yolk syncytial layer, which is present right here, which is represented by YSL. So the deep cells will undergo radial intercalation and is responsible for thinning out of this layer so that this layer can evenly spread outside, or it can evenly outcover this embryo, as you can see in this diagram. And the yolk syncytial layer is responsible for cytoskeleton movements and microtubule driven contractions. So this yolk syncytial layer will help pulling down this blue colored enveloping layer, and with these three components will simultaneously help in the process of epiboly in case of zebrafish. Let's study emboly. Emboly represents the inner movement of the cells from the surface of embryo. So the movements will always toward the interior side, and the processes which are involved in case of emboly are induction and polarity formation, initiation of inward movement, there will be migration in some cases, and at last there will be germ layer formation. Let's start with invagination. Invagination we call in punching. It is the process of inward folding of sheet of cells. As you can see, there is inward folding, and at last there will be formation of this cavity which we will call archenteron. So it involves the inward bending. This is specific inward bending of sheet of an epithelial sheet, and there will be formation of this archenteron. This process is most commonly seen in case of sea urchin endoderm. So how invagination takes place? First there will be initiation by apical constriction. There will be formation of depression. As you can see right here, the depression is forming. There will be inward bending which can be seen right here. The bending will start deepening, and elongation of this inward protrusion will take place, and at last there will be formation of archenteron. The same process is shown right here. In case of involution, there will be interning. So there is one layer, and this cell will start interning and run across this static layer. As you can see, these cells have interned, and these cells will start moving across or moving alongside this static layer, as you can see. So this type of movement where cells will intern internally, we call it involution. The cells will spread inside the embryo along the inner surface of outer cells, or we can say cells are interning alongside with blastocele roof, because this internal cavity is blastocele, so we can call these cells as blastocele roof, and the cells are interning and moving alongside with this blastocele roof. This is mostly seen in case of amphibious embryos like xenopus, and the site of involution in case of xenopus is dorsal lip of blastopore, from where the involution process will start, and involuting cells will form the prospective mesoderm and endodermal cells, and the cells which are migrating are most commonly the external cells. In case of birds and mammals, it starts with the primitive streak and form the similar type of cells. In case of amphioxus, it starts at blastopore lip. So the morphological outcome of the involution is the formation of archenteron, which is similar with this case. The archenteron forms here. Also the formation of archenteron take place, and there will be establishment of three germ layers: the outer ectoderm, the middle mesoderm, and the inner endoderm. And there will be establishment of dorsoventral and antero-posterior polarity. In case of invagination, we say there is in punching of cells, whereas in involution there is a rolling over, or there is in turning of cells. So to what extent these processes takes place? In case of invagination, there is localized depression, whereas involution there is large scale movements alongside the outer margin or alongside the blastocele roof. Okay, so the examples in case of invagination is sea urchin and amphioxus. In case of involution, we have most commonly seen in amphibians, in birds and mammals. Next, come to ingression. In case of ingression, some cells lose their identity and they start migrating individually. So these cells which are represented right here will lose their cell adhesion interactions from other cells. These cells will separate from the epithelial lining, and they detach and start migrating toward the interior of embryo as mesenchymal cells. So this is a perfect example of EMT process, which is epithelial mesenchymal transition, and the movement type is inward migration. They acquire motility and start moving inward. Some of the molecular markers which can be seen in these cells are snail twist, slug, and JAB1 and JAB2 markers. Example of ingression can be seen in case of sea urchins in the process of gastrulation itself, where primary mesenchymal cells PMCs ingress from vegetal pole to form spicules. In case of chick embryo, at the primitive streak formation, there will be some mesodermal and endodermal cells which ingress through the primitive streak. There will be formation of mesenchymal cell populations inside the embryo. There will be establishment of mesodermal and endodermal germ layers. It will help in the initiation of primitive streak. In case of sea urchins, there will be the formation of skeletogenic mesenchyme. If we compare the processes of ingression, invagination and involution, the cell type involved in ingression is individual cells, whereas in both of the invagination and involution there is the involvement of entire sheet. Okay, so invagination in punching of sheet, involution in turning of a sheet of cells, and in case of ingression there will be individual cells losing their identity, losing their adhesive interactions and migrating inward. So the adhesion type is loss of adhesion, and there will be epithelial mesenchyme transition, whereas adhesion is maintained in the invagination and involution. The movement is random or it may be individual. In case of invagination there is inward folding, and involution there is a rolling over. Examples we talked about, that ingression seen in the sea urchin PMCs, invagination in sea urchin endoderm, whereas involution in amphibian mesoderm. The mechanism is EMT and migration. The mechanism is apical constriction, and in case of involution the mechanism is migration along ECM. Now let's come to delamination. Delamination is very much similar to the ingression, but the difference is the cells do not separate. The entire sheet will start migrating inwards. Okay, so a entire epithelial sheet will split up and start migrating like a parallel layer. This is what we call delamination. So formation of two epithelial sheets, one remains superficial and other will start moving inwards. For example, this layer stays and this layer separates and start moving internally. The nature of the movement is splitting and separation of entire sheet, and the cell behavior, cell will retain the epithelial adhesion and adhesive characteristics. Unlike ingression, there will be retention of polarity, and the mechanism is combination of both mitosis and remodeling of junctions. So the outcome will be formation of two distinct germ layers which are ectoderm and endoderm. Examples, if we say in case of humans, the formation of hypoblast from epiblast takes place in this manner, and in case of birds, early blastoderm will split into epiblast and hypoblast. In case of neardians and certain sponges, this process can also be seen. So what is the morphological outcome? So there will be formation of two separate epithelial sheets. Just like in humans, we said that there is formation of hypoblast from epiblast cells. It helps in establishment of the bilaminar embryo disc. There will be two distinct germ layers. There will be creation of primitive endoderm hypoblast beneath the epiblast, which is usually seen in case of humans, and it lays the foundation of gastrulation proper, which is next phase of morphogenesis. If we compare the processes of ingression with delamination, in case of delamination the entire sheet will migrate. In case of ingression, the cells specifically lose their adhesion, lose their polarity, which is maintained by the way in case of delamination, and ingression there is EMT transition. The polarity is maintained, polarity is lost in case of ingression. The adhesion molecules are E-catenin, which are retained in case of delamination, whereas in case of ingression E-catenin interactions will lost, which will lead to separation of individual cells and their migration. Movement types is sheet splits out and give rise to two different sheets at two different places. In case of ingression, we have individual cells migrating. The example is epiblast to hypoblast formation in case of humans. Ingression, we studied sea urchin mesenchyme transition and primitive streak cells in various organisms.